Hello, welcome to our monthly Science Talks webinar. My name is Lindsay Shula, Assistant Director of Development in the Faculty of Science. We acknowledge that the University of Alberta is located on Treaty 6 territory and respect the histories, languages, and cultures of First Nations, Métis, Inuit, and all First Peoples of Canada, whose presence continues to enrich our vibrant community. The presentation today, Keeping Hives Alive, is about 30 minutes in length, followed by a question period. We will be collecting questions throughout the presentation. Please type your questions into the Q&A box, which will appear at the top or bottom of your screen. At the end of the presentation, these questions will be shared with the presenter to address. A video box of the presenter will appear. If it happens to obscure the presentation material, you can move it by clicking and dragging it to another area of your screen. It is now my pleasure to introduce Frederick West, Acting Dean of the Faculty of Science.
Thanks a lot, Lindsay. And I'd like to thank each and every one of you for joining us this month for the Science Talks webinar. It's really great to see people from all over North America coming together to hear this really interesting talk, which I'm very much looking forward to. So it is my pleasure as Acting Dean to introduce our speaker today, Olav Rueppell. So let me tell you a little bit about Olav. He is an evolutionary biologist studying the sociality of honeybees and other social insects. And he's trying to understand the causes and consequences of social evolution across levels of biological organization, from molecules to societies. Olav studies honeybee biology to gain insight into some fundamental questions, such as what influences the structure of genomes, how complex social behavior is organized, and why organisms age at different rates. We had a poll question on that topic. His research is seeking to identify sustainable solutions to improve honeybee health by undertaking and improving the bees' natural defenses. And we're going to hear a little bit about that today. Olav joined the University of Alberta in 2020 after spending 17 years at the University of North Carolina at Greensboro in the US. He's originally from Germany, and he received both his undergraduate and PhD degrees from the University of Würzburg in Bavaria, with a dissertation on queen-size dimorphisms in ants. After that, he moved to the University of California at Davis for additional postdoctoral training, where he began his interest in honeybees, their behavior, and their genetics. He's trained more than 100 students, published more than 100 articles, and he's been funded continuously from a variety of sources since beginning his independent career. Olav, welcome, and thank you very much for joining us.
Yeah, hello everybody, and thank you, Fred, for the kind introduction. It's really my pleasure and my privilege today to share with you some of my research, but also some of my enthusiasm and fascination with honeybees and other social insects. So I'm going to share my screen here and hope you can all see this. And I'm going to turn my pointer into a laser pointer so that I can point to things. So honeybees and other social insects, I find truly amazing creatures, and they have, since the very early stages of my career, grabbed my attention. And when I say honeybees and other social insects, I mean basically 4 major groups of insects that are social and having colonies and organization in groups that cooperate together to achieve common goals. And those are the honeybees and some of the other bees that we are knowing, but they are also including all of the ants, all of the termites, and many different wasp species. So you will probably think about that and say, okay, well, I know a lot about these things because they are nuisance organisms potentially in my kitchen or other places. But what really sort of attests that too is that social insects are amazingly successful. They are one of the big success stories in evolution, that cooperation and division of labor and specialization of individuals in a cooperative fashion leads to tremendous ecological success. And this added level of complexity in the colony is particularly interesting and fascinating to me. So honeybee societies are made up of 3 principal adult groups, and those are on top here, the worker bee, which is a female, in the center, the queen, and there's typically only 1 queen per colony. And then we have on the bottom, the male drone. And all of these different colony members are doing different tasks. So the queen specializes exclusively on reproduction, and she is typically the mother of all other colony members. While the workers, as their name suggests, are doing a lot of the other work, virtually all of the other work, from building the colony to defending the colony, from gathering food sources to caring for the brood, and all other imaginable tasks that a colony could face. The males on the bottom here have an exclusive reproductive function as well, but they're not laying any eggs as the queen does. They are going out on mating flights once they have matured enough to mate with queens from other colonies, and they play very little function for the colony itself. But by specializing and focusing on particular tasks, these individuals can be more efficient at what they do than if one individual had to do everything. And that is part of sort of their biological success story. I wanted to introduce you a little bit into the biology of the bee. When we're looking into a hive, we see that most of the life of the bee goes on a wax comb. So that you see here in the background, these cells that are making up a comb structure that contains cells which the bees use for rearing their young, their baby bees, if you will. We call it brood or larvae and pupae. And then they also use these wax cells to store food, their principal food being nectar as a carbohydrate source that they turn into honey, and then pollen for sources of protein and lipids. You can see here again that there is 1 queen, and she's surrounded by a retinue of workers that care specifically for her. But you can also see that other workers are doing other tasks here. For example, inspecting this bee here is inspecting the cell because these cells on the very bottom are containing the baby bees or the brood. And if you look closely in these cells, you can see sort of a shadow of a whitish grub, and that is a baby bee or a larvae that develops in these cells. And only when these workers become more mature and older are they starting to go outside of the hive to, let's say, defend the hive entrance, or then at their oldest ages go out and forage for food and bring nectar and pollen back to their hives. So why honeybees are important is a 2-part answer. Basically, one, on the one hand, they are really important as scientific models. Even Charles Darwin already recognized that sort of this evolution of workers that don't themselves reproduce posed a problem to his theory of natural evolution by natural selection. A problem that would be solved only 100 years later, basically by a contemporary of this gentleman, Carl von Frisch, who used honeybees for yet another scientific study. He figured out how they orient in the environment how they find food, and how when they return back to the hive after visiting a valuable food resource, they can communicate with an abstract dance language to their nest mates where to find sources of good food. So that led actually to a Nobel Prize for Carl von Frisch. So there are really important different questions that we can ask using honeybees for fundamental scientific insights. On the other hand, honeybees have, of course, a very important applicable or practical importance, and that is their pollination of virtually a lot of our food sources. So almost all fruits, nuts, and vegetables are insect pollinated, and honeybees are by far the most important agricultural pollinator that we have and rely on. And it's for those reasons that colony losses that we can see in the next slide here are pretty alarming. So what I show you here is over several years, the annual winter losses of Canadian beekeepers, and they vary between 15 and 35%. So dramatic percentage of all colonies is lost every year. And the beekeepers are doing a tremendous job despite those losses to be in business and keep being in business. And in fact, trying to build up the colony numbers here that you see sort of as this yellow line to meet the demands of pollination. So I want to just emphasize that there's probably no other agricultural or livestock industry that suffers these losses and is still sort of sustainable in business. And the question is, how long is this going to be sustainable for the beekeeping industry? Now, colonies are defended, and they are well defended against certain sources of disturbance, including humans, by this structure. Many of you will recognize this, and hopefully not too many will have personal experience with this. This is a stinger, the business end of a worker bee, if you will. And for me, this is a professional risk, of course, but hopefully you haven't made too many acquaintances with bee stings in your life, because it is quite painful. The problem is that the honeybees cannot defend against some other threats. And the main biological threat you can see in this slide here is a parasitic mite called Varroa destructor. This mite here in brown is riding on the head of a bee, and scaled up to a human size, it would be approximately the size of a pizza. And this mite is sort of feeding on the bee, riding around on it. And importantly, while it is injuring and weakening the bee, it is also vectoring some deadly viruses. And there's no stinging against this invader. Now, just to show you how accurately Varroa destructor is in tune with the honeybees, I'm showing you this graph that shows how the reproductive cycle of these Varroa mites, as the little brown specks here, is in sync with the developmental cycle of honeybees. So this is basically the beginning of a new bee here as an egg laid by the queen into the cell on day 1. The egg takes 3 days to hatch into a little larvae, a whitish grub, that gets fed by the nurse bees continuously for the next 5 days and grows very rapidly into a full-sized larvae. And just before that larvae is then actually sealed into its wax cell to metamorphose or to change into an adult bee, just like the metamorphosis from a caterpillar into a butterfly, the Varroa mite is actually invading the cell, jumping off of one of the visiting nurse bees and hiding underneath the brood to then exploit that brood and feed on that brood while it is sealed in and helpless at the mercy of the Varroa mite. The Varroa mite needs these food sources and these nutrients that it gains to then reproduce on its own. And the offspring emerges with the emerging bees after 21 days typically. And you can see that a single foundress can easily produce 3 or even 6 offspring, building up very quickly mite populations that are leading to colony collapse if not treated. And so that is a serious health problem for the bees and the beekeepers. The natural host of Varroa destructor is actually not the Western honeybee that we use here, but the Eastern honeybee occurring in Asia. And it has evolved natural immunity, which you can see here. In the top row, you see brood items that have been attempted to be parasitized by Varroa. And these brood items just die. So they undergo what we call social apoptosis, basically to prevent Varroa reproduction. It's a sacrifice on the individual level, but at the colony level, you have still a lot more healthy brood emerging, and you're preventing these Varroa populations to build up. Now, our Western honeybee, Apis mellifera here, that we use for beekeeping purposes, also has some behavioral defenses. And my research program is trying to understand these defenses and then strengthen them to improve honeybee health in a sustainable way. I'm going to show you the central key aspect of this, which is a behavioral defense called hygienic behavior. I have a little video here that I'm going to just try to play for you, showing you 4 nurse bees in the center of the picture cooperating to actually open up one cell that is Varroa infested. And after these bees have opened up that cell, they are removing the brood item in white here that you can see and are interrupting the reproductive cycle of the Varroa in this fashion. The brood item, of course, is dying in the process. So again, it is sort of an individual sacrifice, but it helps the colony health in the long term, and the colony overall will be healthier for that individual sacrifice. Now, this hygienic behavior is performed by highly specialized nurse worker bees. And you can see this in this graph, which comes from one of our studies, where we looked at who is actually performing these different behaviors. I told you that their first act of hygienic behavior is actually uncapping or opening that cell. You can see these gray circles represent individual bees, that there are some bees that are specializing on uncapping behavior, and that's almost all they do. They're true elitist specialists. And on the other hand, we have bees that are participating in the removal of the brood item. And those are not the same bees, they're different, but they're also belonging to this elite specialist workforce in the colony that is performing hygienic behavior. Now, the question is, how do they know which cells to open and which cells to remove, given that all these cells have the same kind of wax capping on top of them, which you see here, basically. And also, I want to remind you that in a typical beehive, it is dark. So the bees can't see anything that might distinguish infested from non-infested cells. So if you think about sensory modalities and what we can use as sort of communication mechanisms there between the brood and the nurse bees, chemicals come, of course, into mind. And so we postulated that there was some chemical communication between the brood that is infested with Varroa mites and the nurse bees that are receiving a signal and then acting upon it. And this hypothesis was supported by early data that we gathered showing that different types of brood are removed at different rates. In different years here, you see this, the effect is slightly different, but the brood type matters. And so that really reinforced our view that the brood is playing an active role here in emitting some form of signal. And so we went on to search for that signal by looking at and comparing the chemicals that are on the surface of mite parasitized brood and non-mite parasitized brood. You can see basically we gathered from a capped brood comb here some mites that were, some larvae that were parasitized and some larvae that were healthy. And we washed the chemicals on the surface off and rinsed them basically in a solvent called hexane. And then we used these different kinds of hexane extracts or rinses to test their biological activity in a behavioral assay, but also B, then to chemically analyze the rinses to see what was actually in those rinses. And just to show you the results here, the mite-infested brood extract leads to a much higher rate of uncapping behavior of otherwise completely healthy cells than the control extract. So there's biological activity. And the chemical analysis showed sort of that there is the most activity by one of the many different substances that are in these rinses, and this is called triacontin. The details don't matter, but it is a waxy substance that is relatively long and stable, emanating from the surface of the brood, then slowly moving towards the outside of the cell. We wanted to confirm this activity and gathered by observing bees in an observation hive, brood items that were in the act of being opened and removed, and compared the cuticular hydrocarbons, or the chemicals from their surfaces, to a control that was Varroa infested but not removed, and a control that was not even Varroa infested. And so you can see again in this graph here on the right-hand side that the quantity of this triacontin was much higher in the cells that were actually uncapped, confirming its role in this hygienic behavior. We then teamed up with some chemists that synthesized this compound to see that the artificial compound would also have this biological activity, which you can see here in terms of rates of uncapping or hygienic behavior, depending on a little bit on the concentration. And it's much higher than control substances that are chemically similar, but not the same substances. And while we were at this study, we tested a few other compounds that were candidates as well. And you can see that we found similar results with these other compounds, which led us then to propose a refined model of this communication, in that there are some more volatile or smaller molecules that are attracting these specialized nurses into a general area that has a problem in the brood. And then we have these larger compounds like triacontin that are not so volatile and help the specialist pinpoint the problem and open the exactly correct cell. So based on that and these results, we made a mixture called the unhealthy brood odor mixture, or UBO mix. And this contained these 4 chemically similar but not identical compounds. And our goal was to make this mixture into an assay to assess the hygienicity of honeybee colonies. And this assay result then we hypothesized would facilitate management and selection decisions for beekeepers. So you can see here, we use this basically mixture to spray onto caps of cells in a circular fashion, and highly hygienic colonies would almost uncap all of these cells that have been sprayed, whereas colonies that are not so hygienic wouldn't open any of these cells. And so this UBO assay was tested and shown to actually predict Varroa infestation level in colonies if the colonies were found to be low in the UBO assay. They had a lot of mites. If they were performing high, they had very few mites. And this was probably due to Varroa mite removal, hygienic behavior, which was also different based on whether the colonies were low performers or high performers on the UBO assay. So the low performers removed a lot less mites than the high performers. And what beekeepers actually care most about is, of course, whether their colonies survive. So we also measured overwintering survival. You can see that the low UBO performing colonies were surviving at much lower rates than the high performers. And these are colonies that haven't been treated in any other way, I should say. So they are really relying on their natural defense mechanisms. Now, the world is concerned about viruses, and so should beekeepers. And these are 2 of the most notorious honeybee viruses, deformed wing virus on the left and Israeli acute paralysis virus on the right. They are simple viruses, much like the coronavirus, and they're causing severe harm to the bees. So we were also interested in finding out whether the UBO assay told us anything about virus levels in the colonies. And it did, as you will see in this experiment. We removed actual hygienic bees that are performing hygienic behavior, and we had a control group of nurse bees that were not performing hygienic behaviors, and then we quantified different kinds of viruses in these individual bees. You can see here that in orange, the hygienic performers had a lot higher virus levels than the control nurses. So the individual behavior, this hygienic removal of diseased brood, is very risky, and it carries a cost for these individuals. But again, at the colony level, it is a beneficial behavior, because the UBO low performing colonies, so the non-hygienic colonies at the colony level, had a lot higher virus loads than the colonies that were scoring high in the UBO assay, or that were highly hygienic. As you can see, the blue bars are much higher for these different viruses here than the red bars. So with those results, we want to follow in different paths. Again, sort of a fundamental question of what is going on in these bees. How do they make these decisions? What are the neurological substrates and genomic consequences of this behavior? And on the practical side, we want to develop this UBO assay so that beekeepers can use it in large-scale operations to predict which colonies might need treatment for Varroa and which ones might not. And also which, if they are selectively breeding for increased Varroa resistance of these bees, which colonies might be good parents for breeding the next generation of bees. So in conclusion, I have shown you sort of that we deciphered the chemical communication that leads to hygienic behavior and some of the chemicals. And that on the practical side, we have now a useful tool for the beekeeping industry to hopefully breed better bees and sustainably improve honeybee health. Now, my other conclusions are that while this work is teamwork, just like the bees are doing teamwork, I could not have done this without a lot of people. Most importantly, my student and now independent researcher, Kara Wagner. But there's also a lot of other collaborators that I need to acknowledge. And all of this could also not be done without research funding. So this has been over the years funded by a variety of sources that I'm grateful for, and I want to acknowledge here. And with that, I'm at the end of my presentation and happy to take any questions.
Well, thank you. Thank you, Olav. It was a super interesting talk. I really enjoyed it. People have been entering questions in the Q&A, and I'm just going to kind of scroll through them from start to finish, and we'll see how far we get. So if it's okay with you, Olav, I'll share these questions.
Sure.
The first one from Andrew Johnson says, I'm a little unclear on something from the beginning. Are all workers females and are all drones male?
Yep, that is exactly correct. So the workers are all females, and there are in a typical colony tens of thousands of these workers, and the drones that have this exclusive sort of reproductive function are only raised during the mating season. There are typically a few hundreds in a colony, and they don't contribute at all to sort of the functioning of the colony, other than being mating opportunities or opportunities for the colony to pass on their genetic material by mating of those drones with another queen. And when I say this, in some school outreach talks, for example, the boys in the class always are very happy to say, yeah, the drones, the males don't have to work. And then, I quickly follow this up by saying, well, after the mating season, they're kicked out of the colony because they're just a waste of resources, and the workers just kick them out and leave them to die And then the girls are like having their heyday with the boys in the class. But yeah, it's a complicated, and maybe you could say, well, not fair, but it's a complicated system.
Righto. Okay, Sheila Campbell asks, is anything known about the genetics of social apoptosis?
Yeah, that's a good next step. I mean, it's a good question and an interesting next step. What leads those individuals to sacrifice themselves? What kind of molecular mechanisms are going on in their brain? We've done another study where a lot of bees that we make feel sick. They're not even really sick, but we make them feel really sick. They are just walking out of their hive basically and saying, well, I don't want to be the culprit of the colony going down basically. And so we are trying to use that as a experiment to then say, okay, well, what is actually genetically going on? But there seems to be not a genetic basis for some individuals sacrificing versus others not. It's really sort of the disease status or the physiological status of the bee.
Right. Henry Sveck asks, would the treatment of the hive for mites with formic or other compounds impact the ability of the bees to naturally determine which cells need to be opened?
Yeah, so I sense you're a beekeeping experience, and I brushed over this and didn't mention at all. There are, of course, some treatment options against Varroa mites that are more conventional, and these include hard chemicals that have certain disadvantages, not in the least that the mites can evolve resistance against them and have evolved resistance against them, but they're also pretty toxic for the environment, the applicator, and lastly the consumer. And then there are so-called soft chemicals, and that's fumigation with organic acids and other organic compounds. And formic acid is one of those. And formic acid is pretty corrosive. The bees can tolerate it better than the mites. And so it's a useful tool in our arsenal to combat these mites. And we have not, to address the question, we have not done any studies to see whether it actually compromises the sensory capacity of those bees to home in on and perform their hygienic behavior. It's a good question. And my guess would be yes, because of the sort of aggressive nature of those chemicals.
Thanks. Craig Scharf asks, can we possibly select for colonies that are good at detection of infected cells? And I think you've sort of danced around this already, but let's see if...
Yeah, it's an ongoing process. So there are actually multiple selection programs all over the world that are trying to improve the sensory capabilities of those nurse bees. From our perspective, it might also be useful to select better signalers because we know that the brood is signaling. So maybe we can also take the other side and see whether we can select for better signaling. But this UBO assay would assess both. It would be just the efficiency of sort of this hygienic behavior.
Great. Maya Evanen asks, is it known if the specialized hygienic bees are more sensitive to UBO than other workers in the hive? Have any electrocentenograms been conducted?
Yeah, we haven't individually compared the bees that are performing the behavior and comparable similar aged bees that are not performing the behavior in context of the UBO assay. But it has been tested with sort of other chemicals and other paradigms. And so there are studies out, which are not ours, that show that they are the more sensitive bees. Yes.
So Helen Fraser asks, what is the effect of the death of larvae, low population of workers?
Yeah, so it depends, of course, on the infection level. So when we're sacrificing a few workers, this does typically not affect the whole colony. Keep in mind that the queen can easily replace lost larvae by laying more eggs, up to 2,000 per day, which is really truly astonishing. And that the investment in that larvae is partially recuperated by the workers cannibalizing the larvae before throwing it out of the colony. So most of the nutrients and energy investment is actually regained at the risk of, also ingesting some of the viruses that are in these larvae. But it's typically not a factor. And I think our overall results that the colonies that are very hygienic overwinter better, which is a function ultimately of the adult population, suggests that overall hygienic behavior is a lot more beneficial than detrimental.
So Olav, are there workers produced during the wintering?
Here in Canada, the winter is long and hard enough that they are stopping complete brood production. In other areas, there is typically a small amount of brood production going on during the winter as well. So if we go further south, and then of course in Florida, you don't have a real winter at all.
Grace McNeely asks, do these issues, mites and viruses, currently contribute to the decline of non-social insects as well?
Yeah, that's a good question. The mites, the Varroa mites, are very specific to honeybees and seem to not affect any other insects. But the viruses that the mites vector and the bees have are not only honeybee specific. Most of them can be found in other bee species at least, and potentially even other insects. But this is an area that, hello?
We can still hear you.
Okay, my monitor just shut off completely, so I just see black, but I can hear you, so this is all
and we
good.
can see you.
Okay, great. So the viruses it's an area that is very much in its infancy. We don't really know yet how widespread they are, but there are initial studies now out that show that these honeybee insects, and also that they can be transmitted, for example, by visiting the same flowers from one insect to another.
Right. Erica Shelley asks, how soon after decapping are you measuring for the chemicals, and could the decapping be introducing the volatiles?
Yeah, so our standard operation is to measure the uncapping after 2 hours. And that is another advantage. Something that I omitted for brevity of the talk is that there is another selection assay. It's called the freeze-killed brood assay, and it involves liquid nitrogen to kill sort of a section of the brood and then see how much hygienic behavior is happening in response to this patch of dead brood. That's not a very specific assay, of course, and it's a very crude assay, if you will, in terms of the signal stimulus, because you're killing a lot of brood. Can you still hear me?
Yep.
Okay. Yeah. So this freeze-killed brood assay it has some disadvantages. It's also not predicting as well colony survival as the UBO assay, and it's also a lot less practical because few beekeepers have liquid nitrogen at their disposal to perform the assay.
Great. Mark Gattel asks, there's a lot of talk recently about light pollution affecting bees, and does this have any relation to their diseases?
Yeah, so in the wake of the colony collapse mysterious disorder that was sort of publicized, which most scientists now think was we actually don't understand what is going on, but the causes are the same or similar to what has been killed colonies before. There were a lot of theories that were floating around, including cell phone towers and radio waves and things like that. And light, I would put light pollution in a similar category for bees, because bees are using the sun and the polarization pattern of the sky for navigating. And there's little evidence that artificial light sources are confusing their navigational abilities. And so I think we can pretty safely exclude light pollution from the list of possible causes of honeybee health problems. And that also, I mean, is supported by honeybee colonies dying in areas where there is absolutely no light pollution.
Right. Okay, Andrea Jeffrey asks, she's wondering if you could elaborate a little more on what causes a colony to be hygienic versus non-hygienic. Can we turn a non-hygienic colony into a hygienic one?
Yeah, good question. Sort of this nature versus nurture question, right? We have done one experiment where we try to train bees to be more hygienic, to teach them, if you will, and it hasn't worked at all. We've also exposed them, just last summer, we have exposed them to a lot of this UBO smell to potentially sensitize them, not desensitize, but sensitize them and make them more aware. And again, this hasn't worked. But we know that selection and breeding works for making more hygienic hives. If you have two parents that are hygienic, your offspring will be more hygienic. And so we think that most of that is sort of a genetic factor or influence.
Yeah, so just a little slower, I guess, but in the long run, perhaps achievable to make the descendants better at being hygienic.
Yeah, the problem is that as beekeepers, we have sort of mostly selected for honey yield and productivity and not for health. And just as in a lot of other domestic animals and plants, that there seems to be a trade-off between the two. So that has generally led to, this domestication has led to less health and less defenses.
They're more vulnerable. So Jim Stewart has a question about solitary bees and whether they also suffer from Varroa, and if so, whether they have any behaviors that mitigate that.
Yeah, Varroa, as I already mentioned, is basically specific to honeybees, so that solitary bees and other social bees don't suffer from Varroa, but they do have the same viruses or some of the same viruses, and there's very little study so far of what these viruses do to solitary bees. You know, there's hundreds of different species of solitary bees, and it's very hard to sort of study every one of them to figure out where the virus does harm and where it doesn't do harm. So in some ways, the honeybees serve as a canary in the coal mine, if you will. And if the same things hold true, then all of our bees should be affected by the viruses, and it's a conservation as well as a practical agricultural concern.
Yeah. So Nicole Piniak was wondering why we do not incorporate Asian bee genetics locally, since they're so good at controlling Varroa.
Yeah, so the Asian bees are a different species, so we can't just interbreed them or cross them in. So that it would be, there are some practical disadvantages or obstacles, not disadvantages, but obstacles, but there are also practical disadvantages because the Asian bees are not as domesticated, if you will. They don't produce a lot of honey, and they are ready to leave their hives at a moment's notice. So beekeepers, of course, want their bees to stay in the hives. Right. These Asian honeybees have sort of the propensity to just say goodbye, we're going to find a different home. And that's not good for practical beekeeping.
Right. Randall asks whether you believe there will be a definitive process for creating bee colonies for Alberta producers that will be healthier and more sustainable, and how long this is going to take, years, decades, etc.
Yeah, that's a good comment and question as well. In fact, we've just started a new project this year to see whether the locally bred bees, so there are beekeepers who are breeding local honeybee stocks, and we're asking the question whether these locally adapted stocks might be performing better than imported stocks. It is true that the Canadian and the Alberta beekeeping industry in specifics are very dependent on imports, and one of the goals is to change that, and despite the short summer, have a supply of queens when the beekeepers need it, and that might be having the benefit of better local bees as well. But we can't answer that question at the moment. We're just at the start of that research project.
So Olav, is the incidence of Varroa and these various viral infections sort of, does it vary quite a bit regionally?
Yes, and you know, I mean, we are so much at the beginning of this that we're dealing not in variants. Now everybody knows about viruses and how variants can differ in human populations. You know, in the bees, we're happy to know even if what kind of viruses are out there. And so it's really sort of, we're at the beginning stages of this. But what picture emerges is that just like the human virome, basically, we are seeing waves of different genotypes and variants potentially. We're certainly seeing a lot of variation between different breeding operations or between beekeeper operations and localities as well as years.
Daryl Delisle wonders if the presence of marker chemicals within a hive could be used as essentially a pest management strategy, you know, identifying that a hive is infested.
Yeah, so that raises the point that I forgot to mention, but almost every single colony has Varroa.
Okay.
So it's not a matter of deciding whether they have Varroa or not. If you're a beekeeper, hobby or commercial beekeeper, you're going to have Varroa, and the only exception for that is New Zealand at the moment in the world. And so it's more a matter of how much Varroa is there. Do they hit sort of that economic threshold where you have to treat and control them with something? And there again, the hygienic bees might be okay without treatment, while the non-hygienic bees definitely need treatment at some point in the year.
So Leah asks a pretty much straight ahead genetics question. Do the queen and the drone have sort of equal impact on producing more hygienic colonies?
Yeah, another great question. And we're collaborating with an Israeli group actually that has also bred for hygienic behavior to answer that question. And it seems that the mothers are potentially more important. But that might be confounded by the fact that a single queen mates with many different males. So this is an exceptional case actually of female polyandry. So where the female mates with up to 50 different males. And so there might be some dilution of the hygienic gene pool in the amount of different drones that are mating with a single queen.
Right, and how controllable is that?
Yes, beekeepers typically have very little control because the queen and the drones fly off into the air and mate there, you know, sort of without anybody watching them. There are possibilities of artificial insemination, but that's very technical, and most beekeepers don't have that capability.
Right. So speaking of beekeepers and their capabilities, Lincoln asks, how would this information relate to non-commercial beekeepers, if at all, backyard beekeepers, for instance?
Yeah, we think that it is equally applicable to backyard or hobby beekeepers, which typically have fewer hives, and commercial beekeepers, which might have 10,000 hives. Our goal is really to develop this UBO assay or spray into something that can be applied by anybody and then assessed by anybody. And hobby beekeepers, often have actually more time to spend with each individual hive and are typically also more interested in not using hard chemicals and sort of being environmentally friendly in their backyard. And so we think that the UBO assay has actually even more potential or is better received by hobby beekeepers potentially.
Great. So besides the mite load, how might a beekeeper determine whether a colony is hygienic? This is from Dustin.
Yeah, well, we hope that the UBO assay is a direct measure that beekeepers can use to measure the hygienicity of their hive. The mite population is a consequence of that. And the mite population, to assess the mite population, there are various different techniques and strategies. So that's an indirect measure, but the mite population also varies a lot across the season. So typically mite populations build up over the growing season or reproductive season because they come out of winter where they don't have any brood. So the mite population is not really big. And then once the bees are starting expanding and raising a lot of brood, the mites have a lot of opportunities to improve or increase their population as well. So the mite levels are actually not an as accurate measure of how hygienic a colony is, even though it is the outcome of that.
Yeah, that totally makes sense. Nicole wants to know, do you see this approach replacing some current approaches for mite control, like apivar strips, oxalic acid, etc., or would it complement existing approaches?
I think we will use, and the beekeepers, when I say we, will use an integrated pest management plan in the end. Because in some instances, even if we have very hygienic bees, we might still have to rely on other controls. But the goal is to reduce these other chemical controls and develop sort of an integrated plan so that the mites are not selected as strongly for developing resistance. Right now, amitraz is sort of the last remaining hard chemical that mites have not evolved resistance. And there are some signs that they are actually evolving resistance to that as well. So there's a lot of panic in the field because we are losing sort of our last hard tool. And so it's almost like a treadmill that we have to get off of and develop alternative strategies.
Yeah.
But it's not going to be, hygienic behavior is not going to be the only solution, of course.
Right. Janet Sperling has an interesting question. Do you have any ideas on whether the hygienic behavior was selected for in the initial selection of the honeybee hives, a founder effect decades or centuries ago? If so, are there some geographic areas where domesticated honeybees have been better selected for hygienic behavior?
Yeah, beekeepers, I mean, this hygienic behavior and the selection for hygienic behavior has only recently emerged. And I should say again that the Varroa destructor mite is also only recently moved from its original host, the eastern honeybee species, to the western honeybee species. So there's only about 100 years of evolution of the western honeybees to this mite. And so hygienic behavior helps also against some other diseases. And so it might be at its relatively low level evolved to other diseases. And that might be different in different places, depending on the frequency of these diseases. But in general, these populations that are commercial have been taken out of the selective natural environment, and beekeepers are preventing sort of survival of the fittest in there because they're treating sick hives, and so they're taking that out of the equation. And now we start thinking, well, maybe we need to put it back in. And there has been several attempts that sort of propose Darwinian beekeeping, where, okay, well, just don't treat your hives, and the ones that are surviving, those ones you can then breed from. But very few beekeepers are prepared to let 95% of their hives die, and then build up from there again.
Well, we're rapidly running out of time. I'm going to do two more, what I think are short questions. The first one from Jacqueline wants to know how rare or how common is it to have hygienic behaviors in colonies? I mean, is it the norm? Are the majority of colonies have it?
Yeah, it's not an all or nothing. So it's a continuum, and most colonies have a very low level of hygienic behavior, and only the special breeding programs are now increasing that level to where it actually helps sustain the hive.
Yeah. And then our last question, since it's 12:55, is from Vera. And she's asking a question she admits is not on point, but still very important to her and important to bees. Is there any hope that neonicotinoids or neonicotinoids are going to be banned?
Well, that's a very contentious point. And obviously there's not only bees in that equation. We think that pesticides are one of the main contributors, but it is obviously there's lots of different stakeholders, and different countries come to different conclusions about those. And I mean, I just want to say that neonicotinoids are replacing other pesticides that are also not really great for the environment or the bees.
Yeah, that's a really good point. Olaf, I want to tell you, I've never seen as many questions as we've gotten today. And unfortunately, there's a whole whack of questions that we will not have time to ask you in real time, but these questions perhaps can be answered after the fact in those cases where there is an answer.
Yeah, I'm happy to do that. And if anybody wants to reach out to me directly, please do. I'm available.
Well, I really want to thank you, Olaf, for a super stimulating talk, and all the work you did answering questions afterwards as well. I want to thank everybody for joining us today for Science Talks, and just a reminder that our next one will be in March. We haven't established the exact date yet, so please stay tuned for more information on when and what for that talk. There will be a post-webinar email that'll include more ways to connect, opportunities for how you can support research and programs such as this in the Faculty of Science, additional resources, and a way to share your feedback. And of course, this talk is being recorded, and so anyone who wants to watch it again or share it or recommend it to someone who missed it, that's certainly an option. So finally, I just want to ask everyone to please take care of yourselves and stay in touch with us.